Hello and welcome to this BioProcess International Ask the Expert webcast. Before we get started, we have a couple of questions that our speaker has asked to learn about from you. So if you can see the center of your screen, there is a question. Why would you consider using CHT resin? What is your primary reason? Please select one. And this will help our speaker understand who's in the audience for their presentation. So again, if you could answer the question on your screen, why would you consider using CHT resin? What is your primary reason? Please select one of the answers provided. It's in my resin platform. I have a unique molecule. I need unique selectivity or something else. Just giving a couple more people a chance to do that. Okay. So the next question from our speaker is, I use CHT resin in a legacy purification process or a new purification process, or both. You can select both if you use it in both. So go ahead and answer the question on your screen if you would. Looks like a couple of you have not answered yet. I'm going to take that as you don't use it yet. Okay. And finally, I'm interested in OPUS columns packed with CHT resin because, and again, select one, your primary reason. CHT is challenging to pack. I need improved operational efficiency. We're lacking column packing capability in-house, or I already use OPUS columns. And again, if you can answer the question there for our speaker, this will really help him understand who's in the audience and what you're looking for. So I am interested in OPUS columns packed with CHT resin because, please select one. CHT is challenging to pack. I need improved operational efficiency. I'm lacking column packing capability in-house, or I already use OPUS columns. Which of those best describes you? Okay, we'll begin shortly. So, hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the chat window on your screen. After the presentation, we'll begin the question and answer portion, and I will ask our speaker your questions from the chat window. Your questions in the chat window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Fletcher Malcom from Repligen. Okay, I'm going to go see. I think there may be some technical difficulties with Fletcher's audio. I'm going to go check on that. If you can just stand by, I apologize. So, it is now my pleasure to introduce our speaker, Fletcher Malcom from Repligen.
Hello, this is Fletcher Malcom, and I'm the chromatography line leader at Repligen, responsible for our chromatography products and product management, mainly focused on OPUS columns. And the subject of today's webinar will be OPUS columns pre-packed with CHT resin. So the agenda today, we'll do a quick introduction of Repligen and OPUS columns, and then get into the data which we're showing today with OPUS columns pre-packed with CHT resin, including pre-shipping data and post-shipping performance data. And then we'll follow up with future developments and any wrap-up questions at the end. Repligen today has been around, Repligen has been around since the early 80s and entrenched in the bioprocessing industry since the mid-80s, and for many years focused on manufacturing recombinant protein A. And this business was a contract manufacturing business, which has is still a critical business for Repligen. And in addition to that, over the last 5 to 7 years, we've built a business with unique technologies direct to customers. And these are noted on the slide here, from OPUS pre-packed columns to Excel single-use ATF systems, cross-flow filtration TFF skids, flat sheet cassettes from Tangenics, and C Technologies, the newest acquisition for Repligen, and we brought in some interesting analytics technologies. So some of these come from development within Repligen as we've invested in new technology development from acquisitions, and others have come over time. So today we'll be focusing on our OPUS pre-packed column line, and as you see some of the numbers on the slide, we continue to grow very quickly. Repligen packs more columns for bioprocessing applications than any other company in the world. This includes columns for lab scale up to the largest OPUS 80 centimeter columns used in GMP manufacturing. And OPUS columns are chosen because they increase process efficiency. There's good process economics that go along with those process efficiencies, and eliminating those packing risks which can occur when columns are self-packed. And Repligen has a complete documentation to support GMP usage. So all of these factors combined, including our flexible configuration model, which I'll outline in the next slide, have generated these generated a situation of rapid growth over the last several years. Specifically, one of the biggest reasons for the growth has been the flexibility in choices that OPUS columns allow, from very large to very small. And this slide shows the complete range of columns and the choices that an end user would have with diameter, bed height, and resin, which makes the columns very platformable no matter what the application. So many choices. And today we're adding a new option or a new capability which gives more choice to end users, and that specifically is CHT pre-packed columns. And so many of you may be familiar with packing CHT resin. It's often difficult to pack, can be difficult to unpack as well, and pack bed stability is generally poor during any sort of transportation method. So we had a goal going back several years ago to develop a CHT packing method which could work in OPUS columns. This was something that end users were asking us for, and we spent some time in development several years back, and more recently went back and spent more time to develop a brand new method, which we launched earlier this year, to enhance the stability during transit. And so the results, which I'll show in the next couple of slides with data, have enabled us to qualify OPUS columns from 5 to 45 centimeter column diameters with this new packing method. And these are available with our standard lead times. We have already packed and shipped a handful of columns with this new method, even though it's just launching or just launched a couple of weeks ago. And the results shown here from one of the earliest customers to receive a column with this new packing method is shown here on the right side of the slide. So you see very consistent results, asymmetry and plate count at Repligen, and then delivery to the customer. So next slide, we have some data showing the internal or our applications data at Repligen where we've packed columns, and this data is all before shipping and looking at asymmetry as well as plate count. So asymmetry on the left, plate count on the right. And you see that the data conforms within the standard spec range that is typically used for CHT columns. And I should note this is all for CHT 40 micron type 1 resin, and all these columns were packed with that particular resin. So some differences, some variants, but this represents sort of a normal range of results that we're seeing with this new packing method. In addition, we have consistent pressure versus flow data compared to the standard reported by Bio-Rad, although we don't know exactly what diameter that Bio-Rad pressure flow line is. We do know it's a 20 centimeter bed height, and these are consistent with the diameters that we've packed all to a 20 centimeter bed height as well. So consistent process pressure versus flow data, very good asymmetry and plate count data, well above or well within the specification range. Now, the most interesting piece about packing CHT columns is the shipping. And so we subjected OPUS columns pre-packed with CHT to our normal shipping test, which is outlined here. It's an International Safe Transit Authority test, or ISTA test. And this test goes through this test schedule listed on the slide, whereby Repligen packs a column, sends it to a certified test facility in the approved packaging, and then the high temperature and high humidity, or the package with the column inside, is subjected to high temperature and high humidity, low temperature, compression testing, 2 hours of vibrational testing, 2 drop tests, and 4 incline impact tests where the box or crate containing the column slides down a ramp at a rate of speed and slams into a steel wall. So that's quite a harsh test, and these tests are designed to simulate transit conditions. And so you can see with the new method, and we're showing a 14 centimeter by 20 column, or 14 by 20 centimeter column here in this slide, the results are very good pre and post. So less than 5% change in both asymmetry and plate count. We repeated this test a handful of times with different diameters. So we have the OPUS 14 centimeter column, which is shown on the previous slide. And then in addition, we've done the ISTA test with also OPUS 25 columns, 25 centimeter columns, and most recently the OPUS 45 centimeter column. And you see the pre versus post shipping data, so asymmetry on the left, plate count on the right. Pre-shipping is in blue, and post-shipping data is in green. The little delta symbol indicates the difference between pre and post-shipping in those bar charts. So you can see that the plate count asymmetry variance is very tight pre versus post-shipping, and consistent with what we would see with other resins. In addition, pressure flow, as you see on the bottom of the slide, is also consistent pre and post. There's no discernible difference between those two. So with this data, we can see that OPUS columns do maintain performance during transit, which is the key challenge associated with pre-packed CHT columns. And with this, we can offer standard specifications, the standard bed height range that we offer in OPUS, and fairly common packing method standards for OPUS columns pre-packed with CHT, which are listed here on the slide. And finally, as we look towards future work, we are gaining experience with our 60 and 80 centimeter columns packed with CHT. There's still a need to look at multi-cycle performance simulation, how packed bed stability performs over time. However, we know from end users that have used the columns sent to date or shipped to date that this has not been a challenge, but it's still something we would like to evaluate. Packing method prospective validation. As you've seen, we've been able to effectively validate the shipping data or the shipping methods for OPUS columns. And so validating the packing method is an additional benefit as we look towards the future and as we increase our volume of columns or number of columns shipped. And then there's some additional hydroxyapatite or CHT varieties. So the new method that we have is developed for the 40 micron and 80 micron types 1 and 2 from Biorad. But there's also some additional CHT varieties which are on the market, and we have yet to evaluate those, but that's perhaps future work as well. And so as we wrap this webinar up, in conclusion, Repligen has developed the first and only pre-packed column available with CHT resin, which has the shipping validation, all the data that I've shown here today, and offers customers a new solution when CHT is required in processing for biologic purification. And so with that, I will close and take any questions. Thank you very much.
Okay, great. And as a reminder, if you just type in your questions to the chat window, I can ask them to Fletcher. So the first question is, are there OPUS columns less than 5 centimeters internal diameter which are available pre-packed with CHT?
Yes, so less than 5 centimeter diameters are available with our Robo column, mini chrome column, and OPUS Valley Chrome columns. And so these are available to complement the larger columns. And the larger columns of 5 to 45 centimeter columns are the ones where we have the new packing method. And the Robo column, mini chrome column, and Valley Chrome columns represent an already established method for those column sizes.
Okay. So recognizing it's a proprietary packing method, can you speak at a high level what the main hurdle was to achieving this output?
Yeah, it's a good question. I think the main hurdle was simply making sure that the column packing results were consistent pre and post shipping, and that and to go along with that, that the packing method itself is reproducible. So it's one thing to say that we have been able to ship a few columns or being able to demonstrate that the columns can be shipped through an ISTA study, and it's another to show that the method is in fact reproducible over different sizes, different size columns, different types of scenarios, different resin lots, et cetera. So with the data shown here, we can be confident that the method is reproducible with those different variables in terms of size, resin lot number, operators, et cetera.
Is there any special transportation, packaging, or shipping methods required for pre-packed columns with CHT?
Today, we're not using any special packaging for OPUS CHT columns, and therefore the packaging would be all the same as what would typically be used for any OPUS column. Although this was something that we thought about and looked at during development, we found that we ultimately didn't need to develop any special packaging, and that the current packaging met the needs of transit and bed stability, or chromatographic bed stability, that is.
So I guess despite all the tests, what happens if the column does not meet specification when it arrives at my facility?
Yeah, so despite all these tests, if a column doesn't, or a column arrives and doesn't meet specification, the first thing that Repligen will do is we'll work on some troubleshooting to isolate the root cause. Why is the plate count and asymmetry not conforming? And so that would be looking at, we would look at things like chromatography test skid setup, making sure that there's no air that's been put on the column. And so with proper troubleshooting, most non-conforming columns can be remedied through things like that. If the column is still not working and the root cause has been determined that it's a packed bed issue, Repligen will replace the column. And so that is part of our standard warranty with OPUS columns, and would also go along with our OPUS CHT offering.
Okay. Well, thank you, Fletcher.
You're welcome.
And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.